My name is Vincent Young. I'm at the University of Michigan Medical Center in Ann Arbor, Michigan. My specialty is in infectious diseases. I'm in the Department of Internal Medicine at the U of M. Today we'll be talking about some of the newer treatments that have been released and are in development for the treatment of Clostridium difficile. Traditionally, the first-line antibiotic agents for the treatment of Clostridium difficile infection have been metronidazole and oral vancomycin. More recently, there's been approval for the optimer compound difficile. When it comes to the effectiveness of the various antibiotics that have been used for Clostridium difficile infection, they're roughly equivalent. There have been some studies that in head-to-head comparison suggest that one of the three agents might be slightly better than the other in terms of the overall efficacy or the ability to stop recurrence. However, there's no real clear advantage of the three antibiotics, metronidazole, vancomycin, and difficile in my experience. Again, some of the studies have suggested that there are some possible advantages in certain cases. But in most cases, the majority of patients will respond to any one of those three agents. Some of the problems that might arise is in patients with recurrent disease, maybe some of the antibiotics might be more effective. But again, none of them are quite as effective as fecal transplantation in the trial so far. One of the newer techniques to try to control Clostridium difficile infection is to actually colonize patients who are at risk for acquiring the infection with a strain that doesn't produce the toxin. As you recall, the Clostridium difficile toxin is thought to be very responsible for the disease that arises. That is the damage to the colon, the colitis, and the diarrhea. There's some thought that if you were to colonize patients with a strain of Clostridium difficile that didn't produce the toxin, it would actually be able to protect the patient if they are exposed to a toxin-producing strain. Because there is some evidence that patients who have recurrent Clostridium difficile infection have this recurrent infection because they do not mount a proper immune response against the pathogen. There is the development of Clostridium difficile vaccines in order to try to increase immunity against Clostridium difficile. In this case, it's not clear if you would be able to prevent a primary infection with Clostridium difficile, but at a minimum, it would be hoped that these patients would not be able to develop recurrent disease, and therefore, if they were treated with one of the first-line antibiotic agents against C. difficile, they would be successfully treated and not have recurrences of disease. For patients that have very severe Clostridium difficile infection, in clinical development, there are actually monoclonal antibodies that are directed against the C. difficile toxin. And the thought here is that in patients with severe disease, again, toxin being the main mediator of the disease, if you can systemically administer an antitoxin in the form of a monoclonal antibody, you would be able to perhaps save patients from colectomy if they're having very severe disease. However, this type of approach is not something that would be thought to be used for primary treatment of an initial case of C. difficile that was not life-threatening.